We are one step closer to the end of our genetics unit, and will now be talking specifically about cancer. Cancer is a disease in which some of the body's cells grow uncontrollably and spread to other parts of the body. It can start almost anywhere in the body, which is made up of trillions of cells. A normal cell simply divides through cell division in a controlled manner, but it's when the division loses its control and we get uncontrolled cell division, resulting in a mass of accumulated cells. This eventually depletes resources and sometimes also invades nearby tissues and organs when it attains mobility called metastasis. Now, masses of cells are also called tumors, which are lumps of tissues. Tumors can be malignant or benign. Some important differences are that malignant tumors are cancerous, whereas benign are not. There is frequent metastasis and no metastasis, and cells have slow to rapid growth, making them more likely to reappear as malignant cancers, whereas benign have slow cell division, therefore have less chance of reoccurrence. Before we begin to understand how cancer cells grow and divide, we will first provide a brief overview of the cell cycle. Now, cells are either in one of two different phases, a phase called interphase, where the cells themselves are growing, replicating their DNA, or just doing their cell functions, or they are in M phase, which is where mitosis, the division of the cell's nucleus, occurs and results in two genetically identical daughter cells. Now, the cell spends most of its time in interphase, and there are a few different subdivisions that occur. The first is known as the growth phase, or G1. As you can see, this is the longest phase of the cell cycle, and here the cells are carrying out metabolic processes such as making proteins to prepare for the next division. Next is the S phase, or synthesis, and it is where DNA replication will occur. And moving on after synthesis is GAP2 or G2, which is where the cell grows even more and prepares for mitosis. Getting to the mitotic phase is extremely important. If a cell has an error, a harmful mutation for example, it could influence the two identical daughter cells that produce and make them mutated. This is where repair mechanisms come in handy. Now, one of two things can occur. Either the cell will take a pause if the problem can be fixed through the use of checkpoints, the most important being the G1 checkpoint, where the cell asks a couple of important questions, being, are we ready to divide yet? Is any DNA damaged? Or is the cell big enough? Or if the problem can't be fixed, the cell will undergo apoptosis, programmed cell death, or self-destruction. This ensures that irreparable cells will not divide. Both daughter cells will become wild types, which means not mutated. But this video will focus on how sometimes these repair mechanisms are not enough, hence mutated cells will result. And in terms of the cell cycle, no gene phase will occur and the new cells go back into S phase, bypassing the G1 checkpoint, and they will re-enter the cell cycle and continue to generate cells that have an error. Hence, this is why cancer results and there is uncontrolled cell division.
Now let's take a look at proto-oncogenes. All cells contain molecules known as proto-oncogenes. These proto-oncogenes are genes that code for the positive cell cycle regulators. They are normal genes that, when they become mutated, are known as oncogenes. These oncogenes cause a cell to become cancerous as the cell progression is much faster. Even if only one allele is overexpressed, a proto-oncogene can still become an oncogene. They code for proteins that help regulate cell growth and differentiation. As protein regulation helps with cell development and growth, the essence of a tumor can cause unregulated cell growth if it messes with the functionality of protein regulation.
The first method we'll look at is gene amplification. So for gene amplification, we start off with two proto-oncogenes. Each proto-oncogene produces an equal amount of protein molecules, which will then go on to help regulate the cell cycle. However, if a gene gets copied multiple times on the proto-oncogene, then that proto-oncogene will produce more protein than the other one. For example, the top one produces twice as much protein as the bottom one. Therefore, the excess protein production will cause the cell cycle to continuously take place.
The third method is missense mutation. So missense mutation changes one of the amino acids of the proto-oncogene, as proto-oncogenes are a form of protein. Proto-oncogenes begin in their inactive state. However, upon the addition of a stimulus, they will become activated and carry out their tasks. If a mutation occurs within an amino acid, then the activated proto-oncogene will become permanently active, and it will be now known as an oncogene. The mutated proto-oncogene will produce active proteins rather than the inactive one. This active protein will cause a cell cycle to continuously occur. So looking at an example of a proto-oncogene, we can look at RAS. RAS acts as the on and off switch for the cell cycle, and it makes proteins that are involved in cell communication pathways, cell growth, and cell death. So here we have a cell, and the cell contains a receptor known as a growth factor receptor. RAS is bound to GDP, The form of RAS that is bound to GDP is considered as the inactive or the off signal for the cell cycle. Until the receptor is bound to a stimulus, RAS will remain inactive. Once the growth factor binds to the receptor, the proteins within the cell will phosphorylate and cause the GDP to turn into GTP, which is essentially the on signal for the RAS proto-oncogene. In doing so, the RAS will phosphorylate a series of proteins, and the last protein to be phosphorylated will enter the nucleus. The nucleus contains a gene that requires a protein, and it will get activated by the protein that just entered and begin the cell cycle. Within a mutated cell, where RAS is an oncogene, the RAS protein is always active. We will still have the growth factor receptor and the proteins, but due to the permanent activity of RAS, proteins will continuously phosphorylate and cause cell production. The regular RAS molecule could still exist and work alongside the growth factor. However, the oncogene is unaffected by this real RAS. Finally, let's take a look at translocation. So, to explain translocation, we'll use two different types of proto-oncogenes, the CDK proto-oncogene and another proto-oncogene that we'll call Z. The Z proto-oncogenes are a proto-oncogene that's always present or is always expressed. During translocation, one of each proto-oncogene of the two groups will switch places, so that we have a combination of one CDK proto-oncogene and one copy of the Z proto-oncogene. Each proto-oncogene has a promoter, represented by P, and this promoter is activated for its specific gene. For example, this one is specific for CDK. However, now that CDK is with a proto-oncogene that is always expressed, it causes a cell cycle to continuously occur as CDK is continually active.
Tumor suppressor genes are normal genes that are responsible for slowing cell division, repairing DNA, and signaling apoptosis. They play a vital role in the development of cancer. Similar to proto-oncogenes, many of the negative cell cycle regulatory proteins were discovered in cells that have become cancerous. Tumor suppressor genes code for the negative regular proteins, which can prevent the cell from undergoing uncontrolled cell division when activated.
Negative regulators such as retinoblastoma protein, RB1, P53, and P21 block the progression of the cell cycle until certain events are completed. Thus, a cell that carries a mutated form of a negative regulator might not be able to undergo the cell cycle. Specifically, mutated P53 genes have been identified in more than half of all human tumor cells. The P53 protein is essential at the G1 checkpoint in the cell cycle, whose function is to monitor DNA. The P53 protein activates other genes whose products halt the cell cycle, providing time for DNA repair, activates genes whose products participate in DNA repair, or activates genes that initiate apoptosis, or cell death, when the damage to DNA cannot be repaired. A damaged P53 protein results in the cell behaving as if there are no mutations. This allows for cells to divide, propagating the mutation in daughter cells and allowing for the accumulation of new mutations. Additionally, the damaged version of P53 found in cancer cells cannot trigger apoptosis.
Sporadic cancer occurs in individuals who do not have a family history of that cancer and who do not have an inherited change in their DNA that would increase their risk for that cancer.
75% to 80% of cancer is sporadic. In sporadic cancer, the gene mutations that cause cancer are acquired and occur only in tumor cells. The risk for acquired gene mutations increases with age and is often influenced by environmental, lifestyle, or medical factors. The most commonly mutated gene in people with cancer is P53 or TP53. More than 50% of cancers involve a missing or damaged P53 gene. Most P53 gene mutations are acquired. Germline P53 mutations are rare, but patients who carry them are at a higher risk of developing many different types of cancer. DNA repair genes fix mistakes when DNA is copied, such as BRCA1, BRCA2, and P53. Many of them function as tumor suppressor genes. Mutations in DNA repair genes may be inherited or acquired.
Carcinogens are substances that are capable of causing cancer. Common carcinogens include tobacco, which contains at least 70 chemicals that are known to cause cancer by damaging DNA, radon, which is a gas occurring in nature but can break down the lining of lungs if built up indoors, asbestos, certain vegetables at high temperatures, which release a chemical called acrylamide, formaldehyde, which is a chemical in many household products, ultraviolet rays from the sun or tanning beds which get absorbed into the skin and damage cells, resulting in skin cancer, alcohol, processed meat, engine exhaust, and pollution.
Cancer can be described as a disease of altered gene expression. Gene expression is the process by which the instructions in our DNA are converted into a functional product, such as a protein. There are many proteins that are turned on or off, known as gene activation or gene silencing, that dramatically alter the overall activity of the cell. A gene that is not normally expressed in that cell may be switched on and expressed at high levels. This can be the result of gene mutation or changes in gene regulation, including oncogenes, specifically oncogenetic transcription factors, which contain proteins that are powerful molecules involved in regulation of cell proliferation, differentiation, and apoptosis. Consequently, the regulator cannot be turned off, which results in overexpression of that particular gene. While these changes don't occur simultaneously in one cancer, changes at each of these levels can be detected when observing cancer at different sites in different individuals, but overall cancer does emerge.
Biotechnology is the integration of engineering sciences and biological studies and has proven to be useful in the treatment of several cancers. The traditional methods to tackle cancer include chemotherapy and radiotherapy. However, both these methods target cancerous and healthy cells as they aim for fast-dividing cells such as red blood cells. There are numerous discoveries in biotechnology that have aided in cancer treatment, one being monoclonal antibodies, or better known as MABs. Monoclonal antibodies are made through cloning unique white blood cells. This was discovered in 1975, and the first MAB was used for cancer treatment in 1983 to treat a patient with non-Hodgkin's lymphoma. Herceptin is an example of a monoclonal antibody that's used to treat breast cancer and stomach cancer, as it is used for cancer that is HER2 receptor positive. A clinical trial at Barcelona Vall d'Hebron University Hospital found that 25% of patients treated with Herceptin went into complete remission, compared to 5% in the control group. Human epidermal growth factor receptor 2, better known as HER2, is an oncogene of which overexpression occurs in 15 to 30% of breast cancers. Monoclonal antibodies only act on a single target. Thus, as soon as one path for the cancerous cells is inhibited, another one takes its place. Solutions to this include bispecific antibodies and the combination of two antibodies. Bispecific antibodies have one body but can form ligands with two different target sites, or the combination of two antibodies can work together. Monoclonal antibodies are a form of immuno-oncology. Instead of targeting the tumor itself, immunotherapies act on the body's own immune system, enabling it to recognize the tumor for destruction. They provide long-lasting memories to the immune system to repeat the appropriate immune response and reduce the risk of relapsing should the same cancer return. MABs such as trastuzumab or Herceptin block signal pathways, killing cancerous cells, while MABs such as ipilimumab trigger the immune system to defend itself against cancerous cells. Cell therapy consists of using a whole cell as a drug instead of small molecules or antibodies. In the last decade, hematopoietic stem cells, which are undifferentiated blood cells from bone marrow, have been commonly used. The standard procedure to treat blood cancer involves taking healthy blood stem cells from bone marrow for modification. Then, chemotherapy is used to blast the immune system, killing off all cancerous and healthy cells. The stem cells from the bone marrow sample are grafted into the patient to try and replenish the system. Gene therapy reprograms a virus into an anti-cancer agent. It involves using specific nucleic acid sequences that once vectorized, which means to insert into a transporter, and delivered to target cells in patients will be inserted into cells, genomes, and expressed as any other gene might. The gene can then be used to activate a certain signaling path or be translated as a protein with many applications. There are two types of gene therapy. One type uses viruses such as adenovirus. An oncolytic virus is used to induce cell death in cancer cells and trigger the immune system or active cytokines. Or a functional group can be introduced which is inhibited in the cancer cells to trigger the immune system. For instance, the p53 gene, which is inhibited in cancer cells. Another type of gene therapy uses a range of non-viral microparticles such as lipofectamine, which is a type of liposome which can freely pass through the cell membrane and deliver the target gene, which is inserted into the bacterial plasmid.